Thank you, Noah and Ariel, for inviting me to come here and talk to you all. I think I've been at NeuroHackademy off and on every couple of years, and so it's always happy to come back and see the new year's participants. And I'm glad that it looks like you have the virtual setup working pretty well too, and so people can participate and see some of the content remotely, and that's always really nice. I'm kind of curious to hear from some of you about how that's been going, because I think all of us are getting used to running these hybrid events and trying to make them as seamless and as engaging as possible, hopefully. Okay, so I'm going to be talking about neurodecoding in the wild. And I have slides, but I do not really care to get to the end of my slides. What I'm much more interested in is having a conversation with all of you here, as well as everyone on Zoom. So Ariel, thank you for keeping an eye on the Zoom. And so I'm happy to take any questions, interruptions as well. Like I said, like my best case scenario is that I don't just stand up here and talk. Please interrupt me, just holler at me, either raise your hand or don't heckle me, but you can yell at me. That's okay if you have questions or comments. Okay. All right. So I'm going to start by talking a little bit about the brain. Okay. You're already here at NeuroHackademy, so obviously you all care about the brain, right? And the brain is a really wonderful thing. So here's a variety of different pictures of different kinds of mammalian brains. On the bottom here is a human brain. And we all have one of these. I'm rather attached to mine, so hopefully you're attached to yours as well. And this is a really magical thing that we want to understand at many different levels. And understanding the brain, whatever that means, sometimes it means something different to each of us, right? So neuroscience is interesting. Neuroscience, we're interested in understanding the brain and behavior at certain different levels. And there's just a lot of complexity to it. And so I just want to tell you a little bit about why I think this is a problem that is both interesting to tackle as well as extremely difficult. So one way of looking at it is that what makes the brain really interesting and also difficult is that it has multi-scale structures in space. What I'm showing you here is on the left-hand side, in log scales, the different size scales that are important for understanding brain and nervous behavior. So each tick mark on the left-hand side is 10 times bigger, going from the smallest size scale to the biggest time scale that I find to be important for neuroscience. This levels diagram was popularized by Terry Sanowski, who first drew it this way several decades ago. And so on the first size scale, we have the synapse. Okay, and the synapse is the kind of the basic unit of connection between one neuron and another neuron. And here I'm showing you a picture of a chemical synapse. And of course, if you think about a single synapse, okay, we have approximately 86 billion neurons in a human brain. Each neuron makes approximately 1,000 to 10,000 connections to other brains. So there's a lot of these things in a human nervous system. Each one of them is already very complicated. Okay, so over here, I know it's too small to see, but there's a lot of people, big communities of big conferences with many more than this number of people in the audience, make their entire career studying what happens to all the proteins and molecules in a single synapse. Okay, this thing has the complexity of a small city, maybe not Manhattan or London, but a smaller city. It's got super highways, it's got garbage collection, has recycling plants, right? It doesn't quite have preschools, but neither does Seattle. So that's okay. And so the complexity of one of these connections in the nervous system is already really complicated, but that's not all, because if you go up in size scales, there's really interesting things that are happening at each of these time scales. So you have the dendrites, right? So the morphological computing through the shapes of the various dendrites and axons that has computational implications. We have entire neurons, of course, which fire action potentials or not. There's layers of cortex, if you're interested in cortex. And then we have nuclei and we have brain lobes. And then you have the entire brain, which of course is not a thing in itself, but rather has the purpose of coordinating the movement of an entire animal's body. In a human, that happens to be something on the order of a meter, because we're on the order of a meter long. And if you are a whale or a extinct dinosaur, you could get quite a bit longer than that, quite a bit larger than that. Okay, so we can go up another order of magnitude or two, depending on what animal we're talking about. And so the cool thing here is not that you can span all of these different size scales when thinking about brain function, but that every single size scale is interesting and has its own complexities. All right. That's not all, because not only does the brain have multiscale structures in space, it also has multiscale dynamics in time. All right, so on the horizontal axis here, we're once again seeing in log coordinates, every tick mark being 10 times longer, different time scales that are relevant when we're thinking about neurodynamics and function. The shortest time scale that's perhaps relevant is the time scale of an action potential, which lasts approximately a millisecond. Now, once again, if you're interested in protein dynamics, if you're interested in kind of like biochemical reactions that are relevant in different parts of the nervous system and different parts of neurons, then you can push this, even farther to the left. But from a systems and computational perspective, often we're thinking about action potentials as sort of the shortest level of computation. But of course, that's not all, because there's tons of really cool things that are happening at intermediate timescales. There's neuromodulation, there's sensation and movement, there's coordination of speech, right? There's short-term memory. I've been talking for a couple of minutes. And so for you to understand what I'm talking about, you are processing speech information, which requires coordination on the, milliseconds or tens of milliseconds precision for you to understand what I'm talking about. And you're also integrating that information with what I said a couple of minutes ago to form hopefully a coherent picture of what I'm trying to tell you right now. And then on and on forwards, right? There's neurogenesis on the order of months to years. And in adults, there's circadian rhythms, monthly rhythms, daily rhythms of all kinds. There's seasonal hormonal fluctuations. And of course, there's long-term learning and memory as well. Humans live on the order of decades. And so that's a kind of a minimum of what our nervous system has been optimized to do, presumably. And so all of this means that the whole system has multiscale dynamics and multiscale structures. And as neuroscientists, I think it is perfectly possible for any of us to choose an intersection of these size scales and time scales. Just pick any, and you can have a perfectly fulfilling and wonderful career at any one of those intersections. Pick an intersection, and there's going to be like a whole conference at the intersection, right? Great people doing great work. And so I'm posing this not because I'm going to answer this question, I can't. I'm posing it rather as a way of saying that I think one of the really important things that all of us should think about and try to tackle is not simply working at any one intersection of these time scales and size scales, but rather thinking about how we can connect them in a way that makes coherent sense, how we can connect them, and not only connecting it to other people who are working at the same time scale and the same size scale, but at least connecting to your neighbors, right? People who are thinking about things that are happening a little bit longer or happening a little bit shorter or at a different level of complexity and computation. And so these are things that, once again, I have no answers to, but I find to be really interesting and to think about in terms of neurocomputation. That makes sense? Okay. I'm going to ask that somebody ask me a question before I continue.
I have a question. I'm on Zoom.
Hi, person on Zoom. We hear you.
So I have trouble with looking at this on a macroscopic scale, because I think my background is very scientific in the sense where it's like we're very structured in looking at what we're already looking at, and then running the analysis, and then looking at what our hypothesis is and testing it that way. So when combining different modalities, it's which I've also recognized as a very valuable thing. I'm just trying to understand how to think that way in a more not collaborative, but more encompassing way correctly.
Okay, sorry, I did not mean to cut you off. Oh, if there's more, please continue.
Yeah. I think that it could lead to dilution of the data or dilution of our interpretation of the data. And that's what I'm trying to avoid. And avoid is the wrong word, but I hope I articulated myself well.
I think that's a great point you're bringing up. Could you tell me your name? I can't really see anyone on Zoom.
Yeah, my name is Tejasvi.
Okay, that's a wonderful question. I think it's a really wonderful point you're making here. And I think so if you look at the history of neuroscience and behavior as a field kind of widely, and I can make it even a little bit wider to the study of animal behavior as well, there's been a there's been a breathing process historically, going back decades or even hundreds of years, where people started out just making observations in what we would now consider to be almost anecdotal or very informal conversation. You read these old papers and they read like a novel more than they read like a modern, what we would consider to be a rigorous hypothesis-driven paper, right? And you're reading and you're like, what did they even do? I have no idea what's going on here. They're just telling the story, right? And that's how they used to do it, because they would just make these observations and they would make connections. And they didn't necessarily subscribe to the same way that we now feel like is a standard of rigor for asking and answering a question when it has to do with neurodynamics and neural function. But especially as I've been talking to some collaborators and colleagues who've also been doing this exercise, they're like, wow, man, like, yeah, this reads like, I don't know what they did, but every single point that they made when I've tried to replicate it, they were right. And, I've had that experience as well. It's like, yes, they didn't do it as rigorously as we might now want to get it accepted in our journal that we want to publish in, but they had true insights and they were right. We can actually often recapitulate some of their ideas and findings even, but using modern techniques. And I think that's very well worth doing. But the point I'm trying to make here is that I think at some point, in part because of the trend towards quantifying everything, and that is like a little more recent, I would say decades rather than hundreds of years, right? Everything kind of became the standard became controlling everything. Okay, and that's very much what I'm trying to, hopefully by the end of the talk, you'll see a little bit what I mean here too about what it means to do neurodecoding in the wild. Everything became a lot more controlled because we want to control enough of the variables in terms of the inputs to the nervous system as well as the outputs to the nervous system, so that we can actually say something quantitatively rigorous about what's going on, okay? But in the process, I believe part of what had been lost is this idea that most of us don't live our lives in a laboratory, that our brain, in fact, functions outside of the laboratory. And I almost never have a repeated trial where I know exactly what the input was and exactly what my output was. And we cannot reproduce it. And that's just what it is, right? Like that's the system that we're trying to study. We don't necessarily want to study exactly what your experimental model organism does in a box that they can do tens of thousands of times. That's great, and that's really interesting, and that's one of the ways that we study the nervous system, but it is not the only way, because we don't ever know if that actually generalizes to beyond your organism in your box, even beyond your lab, right? So I think there's multiple approaches that are valuable here. And so it's difficult and it requires a slightly different way of thinking. So thank you for asking that question. So it's not, you can't often formulate some of these new questions we like to ask in terms of stale hypothesis, like do a statistical test. It doesn't always work like that, right? You almost have to formulate it more sometimes as an exploration question or an engineering question, where sometimes it works and it does work. And we don't necessarily know why yet, and we can investigate it. And then the other thing that this is happening even in some of the animal behavior literature is that there's now increasingly team science being done, where it's not even just interdisciplinary science, because some of these questions require multiple people from different specializations coming together. There are things like the International Brain Laboratory, right, where it's a consortium of a dozen labs from across the world have decided, okay, we're actually going to try, right? We're going to train the same species of animals on the same task, and we're just going to do it all around the world, and we're going to see, are they different? Because that's not clear, right? And we're not particularly interested in knowing what this particular colony of animals does in this particular city in Seattle. We'd like to know if the same thing happens in London, right? So those are attempts of, like, again, this breathing process, right? Where in the beginning it looks like a lot of philosophizing, and then we made it really controlled and really quantitative. And now I think there's a blend, right? Where we're acknowledging that, like, just having really exquisite quantitative control over every single variable doesn't actually go after the question I'd like to answer either. Okay, I just talked a lot. How does that, did I answer your question, or did I talk about something different?
Oh, I think that does answer my question. I think that I'm very restricted in my thinking, where it's like this parameter, and then we'll do a t-test or a regression or blah, blah, blah. And in that, I end up being more restricted in my thinking, which is what I'm trying to get out of. So, yeah.
I mean, it's like, I'm not at all advocating that we we stop doing that. Quite the opposite. I think it's very important to do it that way, the way that you just described. I think what I'm trying to see here is that, especially with some of the new tools to deal with large data sets, diverse data sets, high dimensional data sets, all of these tools that we're taking from artificial intelligence and machine learning, we can handle more than just the kind of the analytic pathway that you just outlined, right? So this is not an XOR, it's an AND, right? And we can do that as a community. Yes, go ahead.
Because I'm thinking of a study. I think there was a report from Berlin from Multiscale to show that whether you go for a walk for an hour in the city or in the greenery, your amygdala responds differently to different places. And if you study people in the lab and control all of the way that you present people's faces to people in the lab, but you don't think of asking them what would they do to your lab.
Yes, exactly.
So, yes, you have all the things that you can control for in the lab, but you do need those exploratory studies to find out things that you can't control or that are beyond your control.
Absolutely.
You just need to, well, this is where you can learn what else is there.
Yeah, I think that's a beautiful example of exactly that, right? And then that study can also itself be replicated, right? So it doesn't have to be like, for example, did it have to be a walk in the magical woods in Berlin? What if you walk somewhere else? Like, does that still hold, right? And so I think it's not like we have to lose the quantitative rigor, right? But just being a little more creative in the way that we ask these questions, like asking questions that maybe people a few decades ago weren't really asking, but now we can ask, right? I think that's really wonderful, and a trend that some of us have been seeing for the last, especially in the last 5 or 10 years, and that's been really wonderful to see for me. Okay, great. Keep it coming, okay? Keep up the conversation. Okay, so there's a couple of things that I've been thinking about that I hope to share with you all. And these are statements that are hopefully not meant to be controversial. Hopefully these are relatively non-controversial statements. I'm going to tell you a little bit about what I've been thinking about and also what my lab has been focusing on to address each of these observations. The first is that the brain is for behavior. Again, might be kind of obvious, but it's shocking how much perfectly good neuroscience that you can do without acknowledging that the brain actually is responsible for producing behavior. Right, because there's like tons of beautiful sensory physiology that has taught us a lot without actually thinking about, okay, so your brain responds to this visual scene this way, what does it do with that, right? Okay, so if we acknowledge that the brain is actually for behavior, we've been increasingly thinking about natural behaviors, right, such as what happens to your amygdala when you take a walk in the woods, and also thinking about comparative approaches, right, thinking about nervous system function in the context of development and evolution. And how it is that we all got to be holding the brains that we have on top of our heads. It was not trained that way in the way that deep neural networks are trained, right? It wasn't like instantiated and trained a bunch of data. It in fact actually had to develop that way from a single stem cell, right? And it also got to be that way through evolutionary time. So I think using comparative approaches is important here. The second observation is that the body matters. And I think this is also a really interesting field where a lot of more classical systems neuroscience has been colliding with biomechanics. Because a lot of, if you admit that the brain is for behavior, then the brain actually has to control a body, right? And then the physical properties of that body, how it interacts with the physical world, also then matters. Because if you had to control 2 hands, if they were floppier, your brain might have to look differently. If you had 6 hands, your brain might have to work differently, right? These are all games that we can now play, especially in simulation. And so that's been really cool. So here we're thinking, again, not only about systems neuroscience, cognitive neuroscience, but thinking about morphology and mechanics. And also what I've been calling embodied computations. This is a term that I sort of borrowed from robotics. They think about like if they're building a soft body robot that's supposed to mimic an elephant trunk or something, right? The way there's computational implications of the arm being exactly as flexible or rigid as it is at any given moment. And I think the same applies more generally, not just when you're thinking about soft body robotics. There are computations that you can make in the central nervous system, and there's computations that you can very much push the periphery. All right, and there's trade-offs of doing one versus the other. And so the computation of the peripheral nervous system and also the limbs is pretty significant in our experience, especially when you're thinking about the brain producing behavior, not simply computing something and representing something, but actually doing something in the real world. And the third observation is that feedback is central. You look at not just the nervous system, but biological systems at almost every scale, and you see feedback everywhere, right? We all know that the visual system is not feed forward. We started out modeling it that way, but we always knew that this was not true, right? Nobody was delusional to think that was true. It was just easier to do it that way, and it was a great place to start. But feedback is really central in the way that we feedback with the environment, in that neural circuitry has lots of feedback at the level of neural regions and across regions. Even if you go down to the level of synapses or protein circuits and gene circuits, there's feedback at every single level of description. And so I think it's really difficult to model almost any biological system without feedback, okay? And so some of the things that we've been up to address this more directly is by, instead of just modeling sensory systems or motor systems, we've been increasingly doing integrated sensory motor models. And I'm also really excited to be getting into reinforcement learning of natural behaviors as well. So that's been a fun new direction with the lab. And so for all of this, we conceive of the following very simplistic diagram, which is that there's an animal, right? It could be you, it could be a fly, it doesn't matter. And it has actions, which is the only way it has of interacting with the world, okay? And it changes the world, and the world responds. And those responses comes back to the animal as measurables. So these are sensory inputs of various kinds. Now I'm lumping in here a lot of things in the world. The world could be physics, right? So if you're a fly, you're flapping, that interacts with the aerodynamics of the air. If you're a fish, you're swimming, then you're swimming in the water, right? If I'm a terrestrial animal, that every step I take, I'm interacting with the ground, right? So you have to model that physics. And this also encompasses social interactions. So when I'm giving a talk, right, I say something and I can look in the audience and you're either nodding or falling asleep or raising your hand or whatever it is you're doing. Best case scenario, in a hybrid conference, the people on Zoom are also responding. So thank you so much for doing that. Not always for sure. And so that's what I mean by feedback. And this is sort of the conceptual framework of a lot of the work that we've been doing recently. Before I get to the specific project I'm talking about today, just wanted to give you like one slide of all the other stuff that's going on in the lab. We do a lot of sparse sensing and control. This we do for biological and engineered systems using a lot of invertebrate flying insects as models. Really interested in quantifying natural behaviors. This is mostly building methods for doing this kind of tracking, but also figuring out analytic and modeling frameworks of what to do with these natural information quantified later. I have a section of the lab that's typically just doing math, data-driven models of dynamical systems. And what we're talking about today are neural decoders and brain-computer interfaces. Okay, so we're just going to focus on the last topic because I think that is the one that's most relevant for human neuroscience. And Ariel told me that that was an appropriate audience for you all, are an appropriate audience for that part of it. But if anyone wants to talk about any of the other stuff, like I'm more than happy to talk about it. Okay. Awesome. All right, cool. Brain-computer interfacing. Have you all talked about brain-computer interfacing before? Should I do like a very general? Okay, cool. So BCIs, or brain-computer interfacing, super trendy right now, right? Super trendy. When the billionaires start getting into it, that's how you know how trendy it is. But there's really cool science engineering to be done even before all of that happened. So brain-computer interfacing, in general, the idea is that one measures neural activity of some kind. It can be invasive, it can be non-invasive. Neural activity of some kind. And you build a computer algorithm that interprets that neural activity and translates it into something that either interacts with software or hardware. So it could be moving a cursor on a screen so you can click on things. This could be an assistive device. It could be operating a robotic arm of some kind, okay? So reading and interpreting neural dynamics that's recorded and measured, and then building some kind of algorithm to operate either software or hardware for whatever purpose that you define. That's like, as far as I'm concerned, if it has those components, it falls under the general umbrella of BCI, brain-computer interfacing. Why would you ever want to do such a thing? Besides that, it's kind of awesome, okay? So the usual reasons that most of us do it are threefold. One, is to assist individuals with physical and neurological disabilities. Sometimes through to a disease or other kinds of disability, individuals may lose the ability to do things like move their arms, to walk, or to be able to interact with a computer, right? Maybe they don't have the coordination to do it with their hands. And so these are rehabilitative devices that assist individuals that need help with a function that they no longer have that they would like to have. So that's one goal. The second goal is to augment engineered systems with humans in the loop. So this one is more like if you think about those surgery robots, right? Where the clinician or the surgeon might interact with a robotic device that augments their ability to do something that maybe they could do with their own hands, but maybe the robotic hand has more precision, or it could be telerobotic, something like that. People also think about this in terms of human operators that interact with lots of different robotic devices, like you can operate a vehicle or something like that, right? Or a drone, or one of those little quad rotor things that fly around. So you can think about augmentation. So for this one, the goal is usually to do better than what you can do with a joystick, because people get really darn good at playing video games. My son is really good at playing video games. And so the joystick is a perfectly viable, very, very good way for us to operate one of these devices. And so the intention here is to do even better than that by not going through the thumbs and the muscles of your arm, right? Can we do even better by directly decoding some intention to do something from the brain directly? So this is a higher, I think this is a more difficult, let's say, about that, right? Because you're not just restoring an ability that was categorically absent, you're trying to make something that already kind of works even better, right? So the bar is a little bit higher there in terms of how well the algorithms have to perform. And then the third one, again, this is the dream of people who have way too much money, is that we would like to perhaps one day enhance all of our lives, because we are living in an increasingly data-rich world. All right, so we have, again, perfectly good ways of interacting with our digital devices, like our phones, our laptops. There's now wearables on our all kinds of wearables, right? And so that already does quite a bit. And so one of the goals of BCI is to explore whether or not there's any capacity for direct brain decoding to do even better, so that we can interact with each other, hopefully in a better way. Given the fact that we have all of this data-rich infrastructure. Okay, so these are the goals. We're going to focus mostly on the first one, because I think that's the one for which there is the most research, because that's also the one for which there is the most clear need, right? The other two, you're trying to enhance something. You're not trying to replace something that somebody has said that they needed. It's more like we do pretty well, but in the future, it might be interesting to dream about a future where we want to do more. Great question. Yeah.
Business brings titles but there's some research groups that they don't use only the brain. Sometimes they use I don't know, if it's some PNG or something like that.
Yep.
And I don't know if that could be considered in this case, or if that I don't know if this idea is more conservative. So
yeah. So that's a great point. I err on the side of not nitpicking between the questions. Oh, great. So the question is, does it have to be brain signals, or what if you're using EMG or some other signal from the whole body, right? Which is a good point. I am not in the business of defining terms and to exclude research. I'm usually more inclusive. So as far as I'm concerned, I would include using EMG and other biometric signals that you can measure in this technology. And the reason I would say that is because what do you think is controlling those muscles? Right? The brain. So at some point it does come from the brain. EMGs are much easier to perform. They're cheaper. They're more robust. The muscle is a gigantic amplifier of those electrical signals from the motor neurons. And so as far as I'm concerned, they're just like, if you can do it from EMG, good for you. Great, right? Like it's easier to do that than actually sticking electrodes around or in the brain. So I don't know if this is what what other researchers consider, maybe they would draw a line somewhere where they say this is not BCI. I mean, like I don't care. It doesn't bother me to call that BCI in some way, especially if it's used in combination with neural signals, right? Which is something I'll show you as well. We've definitely played with those kinds of devices where, not devices, algorithms, where we can use multimodal information to augment the brain signals to something else. And not shockingly, sometimes it works quite a bit better because you have more information. Yeah, there was a question over here. Did I see something on the corner of my eye? Yeah. It does, but I guess, right, like I said, if we wanted to have the semantic argument about exactly where the line is between a word and not a word, then we can do so, right? But philosophically, there's a class of algorithms that we use to interpret electrical signals that we record somewhere from the brain. And you can even put EKG into it, right? And those methods, those analytic methods, are going to be distinct from the methods that you use to interpret joystick signals, right? So there's kind of a practical separation where maybe your EMG analysis or EKG analysis is conceptually related to EEG analysis, right? Whereas if you wanted to analyze joystick output from your video game controller, that's just a different set of math altogether, right? And so again, I don't care to have that semantic argument, but I think there is a practical difference in terms of what are the methods that we can use that generalizes across these kinds of electrical recordings and maybe need to be tweaked if we want to do something else. Is that fair enough for now? Okay. Anyone else? This is great. Keep it coming.
I have a quick question from Zoom.
Go for it.
Actually, I am a UWMC employee. I'm very interested in applied math in the neuroscience field, specifically on the clinical side, neurosurgery. And I think that what you've said so far is so interesting. So just to get on the neuroimaging or EEG side of how I can develop this kind of skill set better, do you have any advice on that? I don't know if this is the right space to ask or time. But maybe I can just.
Well, you're already here the half week, so good job. I think I mean, you're already in the right place for that, right? This is a wonderful opportunity to gain some of those skills, and hopefully that's something that you feel like you have already gained by the end of your time here, right? I will also make a plug just in general for there's tons of resources online for all of these things. The one that's probably that I had a very small part in making is NeuroHackademy. I don't know if you've heard of it, but it's an online summer school. And it has a synchronous component that's actually running right now. I think it started like this week or last week or something. Is it right now? It was last week. Okay, cool. But even if you're not in the synchronous component, all of the tutorials are pretty self-contained online. And so anyone can go through them at any point by themselves, or better yet, go through them with a friend. So you have somebody to work with. And that's only to name one of many, many opportunities for doing this kind of thing online. So if you are looking for resources to, especially in a self-paced way, in whatever time you, whatever bandwidth you happen to have for learning this kind of thing, I recommend NeuroHackademy. Take a look. And if you email me later, and if that's not a good fit for you, email me later. And I'm sure, Ariel, you can all make recommendations as well. I think there's a lot of these resources, and that's one of the great things about living in 2022. Okay. Cool.
Thank you.
Great. You're welcome. Okay. So I've defined BCI, but I did not put a box around it. I'm not excluding anything. But here are two of my favorite examples of BCI from the last decade. Okay, these are so on the left is an example from a group from UCSF, and on the right hand side is something that came out a few years earlier, and this is from a group out of Pittsburgh, University of Pittsburgh. Okay, so I'm going to work through what they did. Let's start with the one on the left, which is what this group did. And this is Eddie Chang is the lead researcher and neurosurgeon in this case. They developed a system for speech synthesis from neural recording. They can actually take recordings from the motor speech region of the brain. These are direct invasive, one of those tiny bed of nails, stabby recordings. And they can actually directly decode the person's, what they would have said and output it as a WAV file. It's absolutely amazing. I think this is one of the coolest demonstrations of BCI that came out in the last few years. So that's cool, right? So that's one, the application here, you can imagine would be somebody maybe having trouble speaking or locked in, or any of these scenarios where a person is unable to speak by actually moving their muscles. This technology has the capacity of giving that voice back to them and playing it out of a computer speaker. Okay. The one on the right-hand side was from a few years earlier, from 2013, and this is direct brain control of robotic arm. So the subject here is patient Jan, and Jan is a quadriplegic. So for the decade before this procedure was done, she did not have use of her arms or legs. And she underwent this procedure where, if you look on the top of her head, there's 2 gray boxes with wires coming out of them. Each of those gray boxes has 100 electrodes that are implanted inside her head. And by decoding the activity out of those 200-ish electrodes, she is able to directly control movement of that robotic arm. So this is a picture of her feeding herself chocolate for the first time in a decade. She looks very happy. I would be very happy too if I hadn't been able to feed myself chocolate for a decade. All right. So this is really cool. And so I'm going to tell you a story of the story arc from my research group. And we started working on this in about 2014. Okay. And so the one on the left was not, I mean, they were already working on it. This was a lot. I talked to the first authors. I mean, they were, before this publication, it was a long, arduous road, but they got to something really impressive. Anyway, so here we are in 2014, and my first graduate student, Nancy Wong, joins my lab, and we start dreaming up things that we'd like to do. And so we were really very much inspired by the robotic arm experiment. And so here we are, and we were looking at this very impressive result that they got. And Nancy is a computer scientist. And so she was looking at this picture and we were talking, and she thought about a couple of things that we would like to make better about this picture, right? This is super impressive, really awesome work. We were just dreaming ahead. Like, okay, what are future technologies we'd like to have now that something like this is very much within the realm of possible? So the first thing that we thought about is, how do we take this outside the lab? This only works in the lab. Jan has to go to University of Pittsburgh and then, get herself into this research lab to use this robotic device. Okay. If we actually wanted her to take this home so that she can grab herself a chocolate bar from her kitchen, what do we have to do? What more algorithm do we have to develop? What kinds of analytic frameworks do we have to develop? Because other people are working on hardware, right? We're going to work on software. What do we have to do so that it might be possible for her to do this outside the lab, her and anyone else? Okay. And the second thing we were looking at is that obvious here that she's not the only person in this frame. And Nancy looked at this, was like, wow, there's a whole team of students and postdocs and scientists and clinicians who are part of this team to make this happen every single day. How do we automate the grad student, right? How do we make it so that there does not have to be a graduate student there tweaking the algorithm every day, right? To make it so that she's able to move this robotic arm, because obviously this is great as a demonstration of concept, but it's not scalable. It's part of bringing it home, right? If anyone actually has to make this useful, they can't have a grad student following them around, tweaking it every day, right? Okay, so we want to automate that. And then a couple years later, we also started working on generalization to other individuals, because one of the well-known secrets of the vast majority of BCI research so far is that all of the models are bespoke to individuals. All right. You collect data from a person, and then you make that model work for that person, and you do it as well as you can. New person comes in, you start over, right? And so again, great for proof of principle, not particularly useful if you ever want to use it. What if you couldn't collect enough training data? Hopefully this is something that you either heard about or thought about already in the course of Hack Week. The availability of training data is everything, right? What if you didn't have enough training data? What if you just had too little? What if you have a new person coming in? Can you make it useful for them without training or minimize the amount of training data you need for a new individual? Okay, those are the kinds of questions that we started to ask. And so we're gonna talk about, we're gonna hopefully try to take a stab at each of these questions. Not suggesting we've solved these questions, but these are the motivating questions for the research that I'm gonna talk to you about. Okay, but I'm gonna talk about data first, because this is something that Nancy also started in the time that when she started her research. So what we realized is that the majority of BCI research, including the BCI research that was done here at UW, was done with little pieces of data that usually lasted from anywhere from 30 minutes to a couple of hours, which is what we can usually manage to collect from either clinical patients, or even if you have somebody coming into the lab and you do an experiment with them, you can reasonably expect to keep them for half an hour to a couple of hours, right? Like that's about it. And if you're really lucky, they come back, and so you get a couple of sessions that are approximately that long. And that's what everyone did. And that was great. We realized there was more, because there was an opportunistic data set from the neurosurgery department at Harborview, where there's a set of procedures that our clinical collaborators, Jeff Oberman and Andrew Coe, were collecting, where these are patients coming in for epilepsy resection surgery because their epilepsy happens to have a focus, and it was not well managed by medication. And so this is one of those therapies of last resort. Okay, you don't want to do this, but for some people, this becomes the very last thing that they would try to help them manage their seizures. And so the procedure is the following. One shows up at the hospital on a Tuesday. It's always Tuesdays. It's a Tuesday to Tuesday procedure. On the first Tuesday, the surgeon opens up your skull. So this requires a whole craniotomy. They take off your skull. Sometimes they leave it attached, so it kind of opens up like a hood of a car. And then they do a durotomy, and then they implant a grid or a set of an array of electrodes directly onto or sometimes into the brain. All right. And then with these electrodes in your brain, you sit in the hospital room, similar to this one, and you sit there for a whole week, right? And during that week, the clinical purpose of the procedure is to localize the focus of the seizure. The opportunity is that you're getting brain recordings 24/7. And before we showed up, they were throwing that data away. They were literally deleting it after this was done, right? So Tuesday to Tuesday, the second Tuesday, hopefully they've localized the seizure. The surgeon goes in, takes out the offending piece of tissue, and then hopefully, fingers crossed, the patient's symptoms are alleviated. And then they throw away the data, okay? And it's not just brain data, it's also video and audio data. We have video and audio data of these individuals for at least a week, sometimes longer. Like if they don't have the seizure localized after a week, sometimes they stay for a few days longer or even a whole other week. And during that week, we have continuous brain recordings of them doing all the things that you would normally do in your whole life, right? You have people eating, sleeping, playing on the computer, and having conversations, both with clinicians and also friends and family who come visit. So they're within the confines of being at Harborview Hospital, they're living their lives as well as they could, right? So we're not talking about natural behavior. They're not playing tennis or giving a seminar. They're in a hospital. They're stuck to the bed. Okay, they're not really mobile. They're not walking around. But given that, they're doing all the things that we would normally do in our lives. And they're doing it not because anyone told them to, not because anyone instructed to, not because a grad student said move your arm. When they move their arm, it's because they wanted to. Because they wanted to do something like drink coffee or play with their phone or reach for the remote for the TV, right? And this entire time, we have video and audio, and we also have recordings from usually approximately 100 electrodes in the brain. We have absolutely no control over what they do. We have no control over where the electrodes are. Those are determined for clinical reasons. And so we just go and download that data afterwards, whenever they're done. And this is, to be clear, ECOG, electrocorticography, which is different than EEG, right? EEG is non-invasive, much more accessible, way cheaper. However, the trade-off is in signal-to-noise ratio. And so here, I'm showing you a diagram from a paper from 2012, where they did simultaneous EEG, so extra scalp EEG, and simultaneously also did ECOG, this much more invasive procedure. And so the trade-off is that, yes, it's much more invasive, and we only get data from the clinical opportunities, but what you get is a much cleaner signal. Those large deflections on the top there, where you see for EEG, that's like the person moving their head a tiny bit, right? There's just tons of things that we don't have to worry about because we are already dealing with a recording of brain activity that's much closer to the source. And so those are the trade-offs we're working with. Okay, I'm going to pause here and ask people to ask questions about the data set. Yeah, it's back there. Yep. Yes, please. Why is ECOG more invasive than the EEG? It's because it requires brain surgery. They have to open up your skull, and then they have to break the dura barrier, which is it's a tough piece of tissue that holds the cerebrospinal fluid inside your brain. So when you hit your head, it kind of cushions it a little bit. They have to break all of that, right? So what you're seeing here is on the bottom diagram. It's actually a picture, so not a diagram. Sorry. The green stuff is the edge of the opening in the skull, and it's approximately this big, right? So if you imagine what you're looking at is a person's brain, this part exposed. This process is very invasive. It requires neurosurgery, which has intrinsic risks, right? Anytime somebody's sawing into your head, it's dangerous. And there's risks of infection, of all kinds of not desirable things happening. Whereas the EEG is just, you put some stuff on your hair, inside your hair, hopefully. Works better when people don't have hair. When I teach an undergrad class and we do live EEG demos, I don't tell the students, but sometimes I'll find one student who has very little hair and they get to do the demo, right? It's so much easier when you don't have hair. Like this stuff, it's just like really noisy. Yes. Yeah. Great question. So the question is about longevity of these intracranial recordings, basically, right? Yeah. So pretty good, actually. So a bit of for context, when I did it, I used to do research kind of electrodes in rats and other rodents when I was in grad school, and we would routinely keep them in there for months without problems. It is possible to do. It's not trivial, right? Sometimes you do run into problems, but people now routinely do small and larger mammals, non-human mammals. You can do it in non-human primates for like years. Mice and rats, months is not a problem. Like if you get good at it, there's the possibility of swelling, but there's ways of managing that. For humans, like I said, this procedure, it has a clinical purpose, and so they just take it out after a week or so. For people like Jan from that robot from Pittsburgh earlier, she had those implants. She was supposed to have them for a year. The plan was for them to take it out after a year, but she loved it so much, she insisted they keep it in. So I think in her case, they ended up keeping it in for 2 or 3 years. And then they finally had to take it out because it got gunky. Technical word, I know. I also have other collaborations that I won't talk about today, where we have, I don't know, like almost daily recordings for 3 to 4 years from humans. And so they could be quite stable. And it's very risky, right? Because anytime you have something sticking out of your brain, there's the danger of infection. There's all kinds of nasty things that can happen. But we routinely have recordings from months to years in both small mammals as well as humans. Go ahead. Great question. So the question is about what makes it hard to generalize specifically to other individuals, right? Okay, I'm going to get to it in a little bit. So I'll talk about that part of the project. It's part of the later part of the talk, but I'll just, the super short version is that there's tremendous variability between humans. So your brain and my brain, yes, they're similar in some ways, but they're also pretty different. If you go to the same like atlas coordinates in your brain versus my brain, no guarantees at all that they're going to be the same, right? They might be similar, like visual cortex is kind of in the same place, but at each individual parcellation, there's no guarantee that my brain, my visual cortex in this spot works the same as your brain visual cortex in that spot. So there's quite a bit of individual variability. The second piece of variability that we have to contend with is these measurements. Like I said, these measurements are done opportunistically, so we don't get to control where the electrodes end up, right, in ECOG, because the clinician just puts them wherever they feel like putting it. But even when you do get control over that, so for EEG, usually people are really careful, like, okay, we're going to put them like in exactly the right coordinates. You are still contending with experimental variability because you try, but you don't succeed in putting them where they are, right? And then there's that individual variability between your brain and mine that we still have to contend with. And so generalizing over that is part of the challenge. Good. Another question? $7 years? There you go. Yeah. So it is possible to do quite a bit longer than a couple of days. Yep.
I have a quick question.
Thank you, person speaking on Zoom. I'm going to repeat the question that's here in person, and then I'm going to call on you on Zoom. Is that okay?
Yeah, of course. Thank you.
Okay, cool. Go ahead. Yeah, so there's a lot of BCI research that's done in non-human model organisms, probably even more, for the exact reason you're saying, right? Because you can get a lot more data that way. There are some questions that you can only answer in humans, or if you can answer it in a non-human model organism, you still have to see if it translates to humans, right? So for example, we're going to talk a lot about moving arms. Right, you can do decoding of moving arms in a rat. They have arms and they also move them, right? But did they move the same way we do? Maybe, maybe not. We'd have to find out, right? And on the other hand, there are things that only humans do, like that speech decoding task, right? You kind of have to do it in humans because rats don't talk, or at least they don't talk the same way we do, right? So for both of those reasons, there is a lot of reason to do both BCI research in humans and also non-humans. Okay, the person who was asking a question on Zoom, please tell us what you're thinking about.
Yeah, my question was the difference between DCM, or direct cortical stimulation, or DCS, sorry, and ECOG.
Direct cortical stimulation
Versus
is a stimulation, right? So ECOG is a passive recording. We're not actually stimulating anything. You can, people, other people pass current through the same electrodes, but I will primarily be talking about just recording and not stimulating.
Oh, I see. Okay.
Yeah.
Thank you.
You're welcome. Yep. Yep. Yep, that's right. So the question is, are we concerned that these individuals have intractable epilepsy? Yep, we are. Yes, there's a paragraph I put in every paper. It's just a caveat of this category of research. Yes, we're aware of it, and no, we will not be doing this to non-epilepsy patients, right? Like, this is a clinical procedure. We will not be doing this to you, unless you hope you don't have intractable epilepsy in the future, but assuming you do not, at the moment, we will not be doing it to you. So that's just a thing. The other thing is that we do look at individuals and see where their center of epilepsy ended up. And if they end up too close to where we're studying, we usually exclude them from the data set. And so we have, we do have some criteria for selection of participants, and we try to get ones where, again, because these individuals have focal epilepsy. So whatever the abnormality is, it tends to be in one particular part of their brain. And all we can really do is say, okay, so this is what ended up being the cause of their epilepsy. We're studying over here. And that's the best we can do. Yeah, there was another question in the middle somewhere. Yes, hi. Mm-hmm. Yes. I will not be talking about it today, but yes, we do. The trade-off there is that DBS is usually implanted in different subcortical structures, And so you're looking at different parts of the brain, which are suitable for reading out different things. The other trade-off is DBS devices usually have a much smaller number of leads. So like 4 electrodes rather than 100 or 100 plus, right? So it's just, again, these are all clinical opportunities, right? We look at a data set that we can get, and we think about, okay, so given this, what are some interesting questions that we can explore so that we can take advantage of this data set that we can have access to. Yeah, so you can answer different questions with DBS. There's a current project that I'm sort of involved in right now where they're trying to do something really cool with DBS, which is like a mashup of DBS and what this is doing here, where they're actually sending these folks home with so instead of having cameras of what they're doing in the hospital room, they're actually going to set up cameras in their own home while they're being monitored with DBS electrodes. They're setting that up. It's like a collaboration between a few UW people and UCSF and Berkeley people. But similar concepts as we're doing here, but you'll be doing DBS electrodes. So the recordings will be different. They'll be more like 4, maybe 8 electrodes rather than a few hundred, but it'll be in their home, which is super cool, right? It's like not just whatever they're doing in the hospital room, it's whatever they're doing in their living room. Obviously, they've given consent for this to be done. Yep. Oh, great question. Yeah. Okay. Great question. Okay. What is the experience like for the individual having this procedure done? Is it painful? So the first answer is that there's no pain receptors in your actual brain, so that part's not painful at all. If you look on YouTube, there's actually some really great videos of awake open brain surgery. Just look on YouTube. There's plenty of them that you can watch if you wanted to. And so this procedure is pretty commonly done when the patient is awake. They just have a sterile field on one side and the person is kind of awake on the other side, and they can kind of talk to them as they're doing these recordings or even simulation. This is in fact the type of procedure that do you know what a homunculus is? Like that cortical map of like the upside down human with like the giant hands and stuff on their map. Like that was first made by Penfield doing open brain awake recording. So people have been doing that for a long time because there's no pain receptors in the brain. You can't actually feel that part of it. You do feel pain when they're doing the actual surgery, like drilling and some of that stuff. So yes, like there will be a team managing that. But it's my understanding is that I never watched one live. You can totally see them on YouTube. It is possible to do these when the person is awake. In terms of discomfort, then afterwards, there's the usual post-op discomfort, right? These individuals are also very commonly, they had been on some kind of anti-seizure meds, and they kind of weaned them off of it after the surgery, so that they would have a seizure while they're being monitored. And so there's usually there's discomfort of coming off of those meds and maybe having their pain managed with other meds that they're receiving. So yeah, it's like it's shockingly okay, I guess, given how invasive the procedure is. It's not like they're in active pain. Yeah, but they're probably uncomfortable. Again, this is not like they're not going out and playing tennis and doing their whole life thing. I think the closest approximation that I can think of is, let's say you caught a cold, and you can't go to school, you can't go to work, you're kind of stuck at home, you're like on the couch watching TV all day for a couple of days, like that, right? You're not normal, you're not doing your normal thing, but you're maybe like stuck on the couch for a while. Back there, yes. Yeah. Yes. We don't control it. This is the beauty of this project, right? We don't control it. That either happens or it doesn't happen, and we analyze it afterwards. So yes, we've been actually really fortunate in that a pretty large fraction of the patients that we've approached for consent to do this have agreed for us to use their data after they're discharged. I think part of it is that we don't do anything extra. This monitoring is being done anyway, right? They're already in the hospital for a week. They're already being video recorded for a week, right? Like that's already done. It's being done anyway. And so all we're doing is keeping that data, and we promise to never release any of the private parts of it. But yeah, you see all sorts like that. You see there are people that we don't analyze because they just really, they're kind of sleepy, they're fatigued, understandable. And they just don't do much in the week. And so we have some criteria in terms of which individuals we end up analyzing afterwards, and they tend to be the more active ones. The ones that maybe have like family visiting, so they're talking a lot, or they're like eating and then watching TV and like just moving around and being awake. Like there's a lot of people that just sleep almost all the time, and we just tend not to analyze them because there's not much we can do because we can't see what's going on inside their brain. Obviously, we only see what they're doing on the outside. So yeah, and so we're also really fortunate in that they don't have to consent to do anything extra, right? So there are individuals that don't consent to participating in other studies where it does require them to put in effort to do something, to like play a video game or to answer a question for researchers to come by or stuff like that. For ours, they don't have to do anything. So sometimes they would actually consent to our study and not consent to the other studies because the other ones require them to be awake and do something, whereas us, we're just watching. And yeah, we see all kinds of stuff like we're talking about. We see them having animated conversations with family members. Sometimes it feels a little weird because, as you can imagine, your loved one being in the hospital, undergoing major surgery, it could be a stressful time, right? And so sometimes they're having pretty emotional conversation with family members who are visiting, and that becomes part of the data set as well. Okay, I'm going to take 3 quick questions, and then we're going to keep going. Go ahead.
Let's say other those are not those pictures are not in any practice with that. So it's just one single event of the event of talk with one parameter, and you don't have any more repetition of that function going on. And in terms of maybe eventually getting some robot response to, I don't know, some function, some reaction, something. What would you do?
That's a great question. So if something only happens one time, we can't do much for that individual, right? So we're taking advantage of the fact that the trade-off is that we have no control and no repeatability, but we have a gigantic data set. So we're just looking for things that happen to have occurred a lot by themselves, right? So if they only talk to a family member one time, we're not going to analyze it. But if they're moving their arm like this a lot, then we're going to analyze that. Yep.
Professor, I have a question, and this is from Zoom.
Let's take the Zoom question. Yeah, sorry. Go ahead.
So if I filter out the EEG signal as much as I can, and how different would the EEG signal be from the ECOG signal, and how different would the information that's decoded be?
We have tried. The ugly truth is that we have actually tried doing what we do on EEG, and it was very difficult. And I'm not brave enough. I have friends who are EEG researchers, and I'm simply not brave enough to have tackled it. And so we backed off and just analyzed the ECOG instead, because like there are things that can be done, but it just, analytically speaking, it gets much more complicated. And so I firmly believe that it actually can be done. We have not yet tried hard enough.
I understand. Yeah. Thank you.
You're welcome. Go ahead. I will answer that question. That's on one of the next slides. But hang on to it. There was one more question I promised I would answer. Go ahead. Yes, usually the electrodes, these are like, a lot of people have temporal lobe epilepsy, and so we usually get temporal lobe, and then if we're lucky, we get a bit of motor cortex, which is what I'm going to be mostly talking about. Let's take one quick question because you have not asked a question before. Go ahead. Oh, what a great question. What happens when the billionaires enter the field and the availability of data? So I'm going to ask you for a favor, and I'd love to talk about the ethical side of all of this. Can you just ask me at the end, and we'll talk about all of that? Yeah, is that cool? Let me just show you some of the technical stuff that we've done, and let's definitely get back to your question. If I forget, like, don't let me forget. Okay? All right. So, okay, so that's our data. What we have is now a big data set We don't actually analyze all of these people, but we technically have like 50, 60 people so far that we've been collecting for many years. Like I said, these are ECOG patients. We have usually, the grid is 8x8, so we usually have at least 64 electrodes, often quite a few more. We have video centered on the individual, so we can look at them. We can't see what they're looking at, just to make that distinction. During the day, it's RGB. At night, it's infrared, and we also have audio of the sounds in the room, and it's in stereo. Now, this all sounds like a lot, and it's also not enough because the quality of the audio and the video are clinical grade, which means that it's not the highest resolution video that you can get, and it's also not the highest quality audio. I, at some point, gave up fighting with them because this is just what they have, and they're happy for us to keep it, but adding any more hardware in hardware review was very difficult. So we stopped doing that. So there's some limitations intrinsic to the quality of the data set. The electrophysiology is really good resolution. And I wish we had better video and audio data, but so I'm just gonna tell you what we've been able to do with what we got. This is all started by Nancy Wong, a graduate student that I co-advised with Rajesh Rao. Raj is in computer science here at UW, and Raj is he's like my partner in crime, right? Like he's the collaborator and co-PI and co-author on every single thing I'm about to tell you about. He also wrote a book called BCI, so that was helpful for your writing variants. Okay, cool. So back to your question, how do we annotate what's going on? We started this project in 2014, right? And I don't know if you remember 2014, 2012 is when computer vision kind of totally, like when CNNs and deep learning blew everything else out of the water. 2012 was when it happens, 10 years. Can you imagine how much it's changed in 10 years with deep learning? So in 2012, deep learning became a thing in computer vision. I was like, oh, this works way better than everything else that we've tried. Maybe we should be using this thing. And so 2014, here we are. And we have like millions of frames of video, right? Hundreds of hours per person and many, many, many people. And so obviously we were not going to be annotating this by hand. And so we were trying to figure out this is before, I don't know if anyone's ever heard of or used Deep Lab Cut. That's sort of the easiest, most successful, most well-documented solution for computer vision tracking of things moving around. There's many other solutions, but Deep Lab Cut is one that many people have heard of. There are other options. Back then, none of that existed, and so Nancy actually glued together a couple of homebrew devices. She kind of glued together this algorithm called YOLO and Cafe Heat Map, and so we like did our own Deep Lab Cut. Same idea, just it was a bit homebrewed. And so the idea was that because we had all these videos and you could usually see the upper body of the individual, their legs were usually covered in a blanket and they weren't really walking around anyway. And so we were really focused on tracking the upper limbs and the head. So we were tracking their head, the 2 shoulder joints, the 2 elbow joints, and the wrist joints. That's what we were tracking. And so with a little bit of annotated data, so we did have some undergrads come and annotate a relatively small fraction of the data. And from that, we trained our own neural network algorithms to annotate the rest. But we're really focusing on the upper limbs here. I actually don't have a slide on this. There's a small side project we did where we tracked facial expressions. So whether or not they're smiling, and they're smiling for real, or they're just smiling with their mouths and not with their eyes because they're not. Yes, thank you for that gift. I love it. Yes. So we have a little project decoding happiness as well, but that was a side thing. Okay, mostly we did movement, like upper limb movements. Okay, and so just a little plug here. If you're interested in the data set, we have been publishing our data sets for a while. The first data set we published in 2017, we call it the Agile data sets, annotated joints in long-term ECOG. That only had 4 participants. The new Agile 12 dataset has 12 participants with much better quality everything. And that is now openly available. I'll tell you a little bit more about that later. I just wanted to make a plug because I think there's a lot here that we have not analyzed, and I would love it if other people would like to. Yes. You're too insightful. The question is, did we struggle to synchronize the video with the ECOG? Yes. So much. It took like a year of banging our heads against the wall. It was awful. We kind of did it. We did the best we could. It was really, really hard to synchronize the video with the ECOG. And it required digging into proprietary software and hardware that over which we had no control. And it took at the end of it, we had to hand synchronize a little bit. And we're at the point where I feel pretty good about the data release. The ones in Agile 12, I think we did a pretty good job, but it was not easy. Yeah. You have no control. You don't have like synchronizing pulses. And then we discovered this is a problem because over the course of a 24-hour day, the two of them were drift by a couple of seconds, which doesn't sound like much out of 24 hours, but it's a huge deal if you're talking about like millisecond precision of movements, right? So yes, it was difficult. I think we did a pretty good job. Back there. Do we have just data collection issues is the question. Some, but it wasn't that bad because this is something the hospital took care of and they had a whole procedure that was working for them. There are electrodes that would go missing. Actually have a new preprint out where we tried to address that data imputation problem where sometimes when electrodes just go missing, we can try to impute it and fill in that gap. That's a different project. What would commonly happen is that, like, I think once a day, sometimes they would just unplug the electrodes, clean it, and then plug it back in. And so there'll be artifacts associated with that. We'd be missing a small chunk of the data. But these are all just normal issues with recordings. But yeah, like just recording at all was something that was worked out by hard review. So we were not concerned about that. And we had so many. So this procedure before COVID, on average, this was usually done at hard review once a month. Like there was one patient per month. And so conservatively, let's say that we get 10 patients per year, right? And we were collecting this data since 2014. And so that the quantity of the data was not really a problem. Like if one person happened to have somewhat corrupted data, we couldn't use it, it's not a big deal, we'll just move on to the next person. Yep. Do the electrodes move during clinical procedure? Yes, somewhat, but not a whole lot. Yes. They're kind of stuck. The ones that are extracortical, like they're kind of stuck onto the cortical surface. Yes, they can move. Practically speaking, they don't move a lot, but yes, they can move a little bit. And these are large-ish electrodes, so we're nowhere near single cell resolution. Right, so if anyone maybe have done sharp electrodes or tetrode recordings or neuropixels or something, at that resolution, if anything moves at all, you get different cells. But here, because we're already averaging probably thousands, if not tens of thousands of cells, if the electrode moves a little bit, you're still averaging over the same population. So for us, it's not as big of an issue. Cool. So now we have data, and now we tracked people moving their arms. And at some point, we were able to decode these movements. When Nancy graduated, I had a couple of other people join the team, and they looked at the data and they were like, okay, we need to do better because there were just too many errors. And we wanted to do better. So this is like a ton of heavy lifting by Saprit Singh and Steve Peterson, both of whom work at Meta now. And and so what they did was that they did the thing that Nancy did, but they just cleaned it up and made it even nicer. And so now not only do we have better quality tracking of all of the joints, we also improved electrode registration because for each individual we map in 3D where the electrodes are on their particular brain. We have much better robust movement detection. So we used a model for detecting when the movement started and ended with a lot more temporal precision. And we have characterized a bunch of movement metadata, like not only are they moving, but what's the magnitude of the movement, the direction of the movement, whether or not it's one arm versus also the other arm, if they're moving and talking at the same time. I do that a lot. My husband's partly Italian. I am not, but like I talk and move my hands, a lot. So people do that, and we can characterize all of that as metadata as well. So here's a short video of just 16 examples. And we have like hundreds, if not thousands of these per person, right? And so these are all movements of the right hand, okay? And they're all synchronized to where we detected the movement to start. So it's going to loop over and over. So you kind of see what the movements are. And so I'll point out here that there's a lot of similarities, right? So in each small video clip, the person is moving their right arm. However, there's a lot of variability as well. The first one I'll point out is that there's a lot of variability in lighting. It's Seattle, so usually during the day, the window's open, depending on if it's sunny or cloudy outside, there's a lot of lighting variability, which is challenging for computer vision. So I feel pretty good that we were able to deal with that. You also see that sometimes the movements are occluded, like when they have that tray in front of them, right? Sometimes there's like parts of the arm you can't see. Sometimes they're on top of the tray. Sometimes they're under the tray. There's videos that are taken at night. So this one in the middle here, you can kind of see it's in black and white. So that was taken during the night because it's just a black and white infrared video instead of being an RGB video. You'll see that in some of them, there's another individual that you can see walking around. Sometimes it's one of the clinicians, sometimes it's a family member. And sometimes they're just moving because they're moving. And sometimes you can see that they're actually reaching for something, right? Like in many of these cases, there's a meal or a drink or something next to them. And so they're reaching for something. And they also sometimes scratch their head. That's another common movement that we see. So we can characterize all of these things, right? And what we end up with is, first of all, a really cool data set where we can just analyze the variability of natural movements, right? That's the first thing we looked at, which is, okay, so now that we have all these movements, okay, what's the natural variability here? What you see here is each row is an individual from S1 to S12. Each row is a person. They're all in different colors. We took them over many days of recording, but depending on whether or not they were sleepy one day or really active the other day, we don't get the same number of movements from day to day. Some folks moved a lot, some folks didn't move as much. You see that if you look at the distribution of time of day, it makes sense. They're generally more active during the daytime and less so at night, but not always. And you can see that like some people wake up earlier and go to sleep and stuff like that. And they also do move during the middle of the night because some people do. We can then look at the distribution of things like reach duration, reach magnitude, reach angle, and onset speed. Once again, there's a huge variability both within and between individuals. You'll see that, for example, the onset speed in terms of what's the speed of their movement in pixels per second when they start moving, you'll see that some people just move faster than others, which again makes sense, right? Some people move faster, other people don't move as fast. These are just all variables that we can characterize and quantify now that we have this data set. We can also quantify things like how often are they moving and speaking at the same time? Once again, people are variable. The person in green, S3, is almost always talking and moving their hand at the same time. And some people don't, right? This is the thing, we're all different. And then there's also things like how often are they moving both hands and other things like that, right? We made up a bunch of metrics and we were able to quantify all of this. And so the thing that I want to acknowledge here is that especially when we're doing human neuroscience, I think it's really important to, in this era of big data, to resist the urge to oversimplify and only report average statistics. I mean, it's not just about the mean and standard deviation, right? There's so much more there. And the difference between me and you is what defines us as people, right? Like I value that difference, not just as a statistic, as a number, because the difference between me and you is what makes me, me, and what makes you, you, right? So especially in human neuroscience, but even more, actually this applies. I mean, one of my colleagues does individual differences in fruit flies. They have individual differences too. Just acknowledging that difference as something that is valuable to understand and study rather than a nuisance in your data set, I think is something valuable that we can do. And so that's just the behavior. Now we can characterize what happens in the brain at these movement onset events, right? What we've done here is we've now averaged all 12 participants and we projected all of the electric recordings onto a canonical map and parcellated it out by some predefined cortical areas, okay? And what we see on average is that we can now compare the neural correlates of these natural movements of the contralateral arm and compare it to what we thought we understood before in terms of what happens during instructed movements. So it's been well studied before what happens in the human brain when you move your arm, and these are instruction movements. Like I'm sitting there, I'm a grad student, a postdoc or whatever, right? I'm saying move your arm. move your arm again, you move your arm. You do this like, 10, 20 times, right? And I average your brain activity when you did that. Here, the difference is that the movements are relatively the same because we're still looking at your arm moving. The only real difference is that we're now looking at movements that are not instructed and are spontaneous and are in a naturalistic context. And on average, the short story is it looks the same, which is reassuring, right? It means like things are not completely different under naturalistic conditions. What we see here is in especially the precentral and the postcentral areas on either side of the central sulcus, and this corresponds to what we would normally call S1, primary somatosensory cortex, the M1 motor cortex, primary motor cortex. Everything is pretty sensible, okay? What you see is a general increase in activity. These are spectrograms. So frequency on the vertical axis, time synchronized the onset of the movement with the line is 0 when they started moving. And color corresponds to increase or decrease in power. Red is increase. So you see increased power, what we call the high gamma band. And we see a generalized decreasing power in the low frequency band there. And this effect is consistent for precentral and postcentral, and it gets weaker as you get farther away from the somatosensory and somatosensory motor regions of the brain. So for example, in inferior temporal, which is that kind of that purplish region, right, pretty far away from the sensory motor cortex, you don't see anything at all. And this is good because this is exactly what we expect. Okay, so far so good. Natural movements look a lot like instructed movements. So this is good. On average, if you look individual by individual, it goes bonkers. Okay, here's what happens if instead of averaging over everyone, you look at every individual by themselves. Now, if you look over these 12 folks, you'll see that some of them look a lot like the canonical picture. So let's say S3, S9, S6, S11, S7, like they have patterns that largely resemble what we think we should get based on controlled experiments from previous literature. Okay, they look pretty similar, but some people just don't look like that at all. So S8 does not have that generalized decrease in the low frequency band. That blue band is just missing altogether. S12 barely has any increase in the high gamma. Don't know why. Like these people are just different. Even people that do have the canonical pattern, you'll see that the frequency band at which the decrease occurs shifts. So S1, that blue blob at the bottom goes up higher than it does on average. And then S4, that blue band is lower. We don't know what's up with that, right? So like this is an exploratory project that we've continued exploring. And so what we've done here is tried to at least in an expository way, characterize the variability in these natural movements, both at the behavioral level and the neuroactivity level. Explaining it is something that we did try to do in the paper, but I mean, the truth that I will admit to is that it didn't work nearly as well as I thought it would. We published it because that is what we did, and we reported the results, but we had trouble explaining this variability. So how much of it is explainable in terms of other observables? A little bit, not a huge amount. How much of it is individual differences? I don't know. But this also underlines the difficulties of a generalized BCI, right? So if you really want to generalize to more individuals, we have to contend with this. We have to contend with the fact that your brain and mine might just work so differently that, it's not going to be easy for me to train something on me and generalize it to you. Question there and then there. Yep. Great question. We have a structural MRI for everyone. I'm not aware if we have fMRI tasks for these folks. Yeah. That's a good question. That is a good point. Yeah, I've been pretty interested in doing in analyzing data that's simultaneous ECOG or EEG and fMRI for a long time. We don't have the data set for these particular individuals, even non-synchronously. But there's always like a couple of collaborators who are interested in doing some of this kind of analysis. So I'm really interested. I don't know, but that's a great question. Over here.
Data that comes from or is that hard to do?
So is there a notion of functional localization with these passive observations? I think that is one interpretation of what we have done here, right? We're essentially doing a localization of the cortical region responsible for arm movements, right? So I think that's one interpretation of what we have done. Can you do more? Probably.
Maybe the change of the small before the electrodes were what actually exactly was.
Yeah, so S2, I mean, so we did, these are all projections onto the parcellation. So this is not electrode by electrode. This is like a weighted average of all the electrodes that would have contributed to what would be in S2 if we actually measured S2. So, and we excluded individuals where there was just no coverage altogether. So some folks had like nothing near S2 or S1 even, right? And we didn't analyze them. So this population was already pre-selected for reasonably good coverage of M1 and S1. Yeah. Was there another question? I will keep going. Okay, cool. Oh, it's already 5 o'clock. I've been talking for a really long time. I'm sorry. I can just end. This is what I promised. Come back to the ethical question. Okay, you know what? Let me show you one more thing that is cool. Let's see. Okay, I'm just gonna load this up into your brain because it was something that I started talking about. We now have a way of, I won't show you all the details. Basically, we developed a way of doing generalized transfer learning on decoding, so that we can, like, because it's really hard to train it on one person, generalize to another person. But what we now do is we train on a bunch of people, and we have demonstrated that we can now generalize to another person. And if you can do a tiny bit of fine tuning, so in our data set with 12 people, right? So we train on 11 people, and then we go to the 12th person, sight on scene, and it works, doesn't totally suck. Right. And if we train that model a tiny bit on the 12th person, it does about as well as you can possibly do. Right. So this is a data size problem. Like, we're taking advantage of the fact that we don't have a lot of data for any individual, but we have a large number of individuals, and we can train models that generalize. Yeah. Yes, some people transfer really well and some people don't. Yes. It depends. It correlates, as you may suspect, it correlates with overlap and coverage. So if you have somebody in the data set that has closely overlapping coverage with your test individual, it works better. And the more the overlap, the better it works. Yeah. Okay, so we've done that. And the other thing we started playing with is self-supervised learning. Because again, the dirty secret in all of this is that I, over many, many years, have paid a lot of undergrads to do a lot of labeling for hundreds of hours. And they labeled like 1.5% of the data, right? But that was still hundreds of hours of human effort labeling the data. And so really, we want to get away from that altogether. We have some ideas of how to do this. And the bottom line is that you can take advantage of the fact that we have multiple streams of information. So if you have neural data and EMG data and video data, then they can basically train each other in a way that doesn't entirely get rid of the need to label, but it sure makes it a lot more scalable. Okay. And then this entire Agile data set is published as a Dandy set. Dandy is a public data repository. That's very good and very, very cool. And everything has been put into NWB format, so neural data without borders, a standard data language for describing behavior or electrophysiology data. This is a huge, huge effort, heavy lift by C. Peterson, with help from Ben Dichter and Michael Sheed, who are also Dandy and NWB evangelists, so they helped us. And the whole data set is pretty gigantic, so it's a big commitment to download it onto your actual computer. So we built a browser-based exploratory device so that you can kind of play with the data a little bit and then figure out which part you actually want to download and work with if you felt like dealing with it. And so this is just an advertisement that if any of what I said sounds interesting to you, you want to play with it, you want to use it for your research, everything's public, go play with it. Okay. All right, I'll stop there. Thanks. I feel pretty good about the conversation we had today. So if there's any questions, I'm here, or we can just end. Up to you. Oh, the ethical question. Yes. Okay. Just restate your question again, please. Yeah, so I mean, I'm gonna speculate at a couple of different fronts, because one of the things we've talked about is availability of data, right? And so in other fields of machine learning, the ready availability of data was one of the game changers. So in computer vision, there's now bigger amount of data sets, but the fact that ImageNet existed was a really big deal for that field to collectively make progress forward, because they had a much larger data set that they can all agree on that this is the standard of how we can do better, right? So I firmly believe that collecting larger and larger standardized data sets just as a benchmark for, for my research to be even comparable to yours in an apples to apples way is really key to moving forward, because otherwise I'm just fiddling around, you're fiddling around, and like I say mine works and you say yours works, and it's great, like who knows, right? So it kind of depends on whether or not those individuals choose to contribute to the effort by actually collecting and publishing data, or if they're going to keep it for themselves, right? They can certainly choose to do that. They don't have to share it with anyone. It also depends on how they collect it, right? And what they're doing. The other thing that's the other piece of it is thinking about just like the potential of this technology, right? Because there's a lot of things that we want to do. And what is not in here is which of this is going to make people money, right? So I think like computer vision wouldn't have progressed nearly as quickly if it wasn't obvious how large corporations can almost immediately make money from that technology, right? For instance, at some point in the last few years, I gave one of these talks somewhere, and afterwards I got an email from a researcher from their business school saying, hey, I'd love to analyze this data set and figure out people's responses to commercials being played on TV so that I can better predict how to craft commercials and advertise more directly to these people. And I was just like, I'm not responding to this email. But you can kind of imagine things like that happening, like the fact that we can do this. Yes, we have ideas about what we'd like to do with it. And the commercialization aspect is it's dangerous, right? But it's also good, because if you can commercialize it, then you automatically get a lot more resources, and it makes things possible that really were not possible before. So you have to think about both, right? So like there's only so much all of us academics labs can do, kind of just playing around with whatever we happen to be able to get, right? If you don't have resources, there's a limitation there. And sometimes the best way to get the impact you want is through commercialization. Right. And that's worth considering. Maybe not in a competitive way. I have no interest in competing with billionaires, right? But sometimes the best way to get more research done is to commercialize, get IP, get a startup, right? Like there comes a point. We're obviously nowhere near that for the stuff that I talked about, but there's definitely a role for that. But I think like a lot of how people have gotten in trouble, especially with the whole Silicon Valley tech wave of technology, is the hubris of thinking, oh, we're just going to build the tech, the rest will be fine. The rest is not fine, right? And especially if you're thinking about this kind of technology. So I talked a little bit about project. I didn't show you, but we have a project where we're reading people's emotions, right? You can actually decode. So here we're decoding movements, which is an objective thing. Like you either moved your arm or you didn't, right? You can kind of decode people's emotions. There's our paper, there's another paper based on a USC group. They did similar things, different metric of how they measure emotion, but basically they're trying to decode whether or not the person is happy or not, right? So then you start thinking, okay, so what does this mean? It's cool technology if you can decode whether or not you're happy, right? Okay, so I can decode whether or not you're happy, which means that I can make an app out of it. So you have an app on your phone that tells you what mood you're in that day. Okay, great. That means your partner might have an app on their phone about whether or not you're happy today. Okay, what are they going to do with that information? Right? Especially if you are being treated for something else, right? Where you have a choice of either upping your dosage on some medication you may or may not be taking. If you have the choice of turning off your DBS, right? Like, what are they going to do with that information? Not just you, but the people around you, your friends, your parents, your children, your partners. Like, what are they going to do with that information that they can see on their phone that you're either happy or grumpy or whatever it is that day, right? Do you really want them to have that information? It's technically getting towards possible, but what apps are people going to build and how are they going to make money off of it? These are things that we need to talk about. I'm sure other people have thoughts. Yeah, that's a good one, right? So like, yeah, I had a friend who they didn't, I don't think he was ever, I'm not sure how, he wasn't diagnosed with an autism spectrum disorder until later, but in retrospect, his mom was saying, oh yeah, when you were a kid, you kept asking me, mommy, are you mad? It's like, no, I'm not mad at you, but he kept asking because he didn't know, right? Yeah, like what if you had that on an app, right? This could change a lot of people's lives. Micro expressions. There is, I've thought about this quite a bit. Maybe I'll just end on this note. I've thought about it quite a bit. And it's hard for me to imagine any technology in the history of all humanity that has not been used for good and bad. It comes with a territory, right? Anytime there's an innovation, anytime we build something new, anytime we gain a new capacity, people seem to have great creative uses for it that make lots of people's lives better. And then also people use that same innovation, the same technology to make people's lives worse. And rather than ignoring that that's a possibility, right? We just need to think about it. Like the people who are trying to do good need to also think about, okay, so what can we do that's bad, right? Because the fact that you're not doing it doesn't mean that someone else is not doing it. And we might as well know about it and think about it rather than just waiting for it to happen. So yeah, it's like, I firmly believe that there's a lot of good that can happen here, but it has a tremendous capacity to be abused as well, like every other technology. So, thank you all. I'm especially grateful for all of you in person and on Zoom who asked questions in the middle. I'm glad I asked you to ask, and I'm sure glad that you did, because sometimes I ask and people don't do it. So thank you so much for talking to me. This is really fun.